Hello everyone, welcome to the lecture series on basics of cell biology. Today we are going to learn about most important cellular organelle, that is mitochondria. In this video we will discuss about the structure, that is morphology, chemical composition and functions of mitochondria. So let us start with mitochondria. So what are mitochondria? So if you see these mitochondria, they are known by more than 50 different names. So different scientists have given different names to mitochondria and are the filamentous thread-like or sometimes granular cytoplasmic organelles which are found into the eukaryotic cells. So we know mitochondria, they are only restricted to the eukaryotic cell. They cannot be seen in prokaryotic cells, and they are filamentous, that is thread-like or sometimes granular cytoplasmic organelles, and they can be seen in both, that is plant as well as animal cells. They are absent in prokaryotic cells, and they are first of all described by the scientist Richard Altman in 1890, and at that time he called them as bioplast. But after that, the term mitochondria, it is first of all used for bioplast by the scientist Benda in 1897. Then there was a scientist Nas 1963. He observed and proved that the mitochondria, they will contains the DNA, a special type of DNA molecules, and that DNA, it is known as mitochondrial DNA, or it is known as mtDNA. Also it is known as extranuclear DNA because it is other than the nuclear DNA. Also, it consists of various types of enzymes, coenzymes, also a special type of ribosome, 70 ribosomes. They are also known as mitoribosomes. They have their own machinery of protein synthesis. So they are capable of self-replication, and they also participate in inheritance and differentiation of the cell. So that they are known as autonomous organelles or the cell. So mitochondrial DNA, they works in coordination with nuclear DNA because they individually they cannot work for inheritance. They will work in coordination with nuclear DNA. If we see the diameter of mitochondria, it ranges from 0.5 micron to 2 micron, and length may ranges from 0.3 micron to 40 micron. So this is the diameter of mitochondria. The mitochondria, they are concerned with the synthesis of adenosine triphosphate ATP, and which are the chemical form of energy. Hence they are popularly known as the powerhouse of the cell. So they are known as powerhouse of the cell. Now we will see structure of mitochondria. So what is structure of mitochondria? If you see this mitochondria, or the number of mitochondria in the cell, it depends upon the different types of cells or the functional and physiological state of the cell. So number of mitochondria per cell, it varies from cell to cell, and the range, it may be few hundreds to 5 lakhs per cell. So in some cells their number will be few hundreds, and in some cells it may be up to 5 lakh mitochondria per cell. The number of mitochondria, it is lesser in plant cell as compared to animal cell. So mitochondria, or their number, it will be higher in animals than plants. There is hardly any structural difference between plant and animal mitochondria. I mean, structurally they are very similar in plants and animals. The shape of mitochondria may be different in different cells based on the physiological conditions of the cell. So in some cell they may be filamentous, in some cell they may be granular, in some cells they may be club shaped, racket shaped, vascular, or round in shape. But typical mitochondria, they are having the shape of sausage. Now under electron microscope, if you see, observe, mitochondria shows the different parts like mitochondrial membrane, mitochondrial matrix, and mitochondrial particles. So the mitochondria broadly divided into these three different parts. So first and very important part, it is mitochondrial membrane, second is mitochondrial matrix, and third is mitochondrial particles. They are also known as subunits. So entire things are given here. This is the three-dimensional view of mitochondria. This is the a sectional view of mitochondria. If you see, these are the mitochondrial membranes, and they also contains this matrix. So this is the mitochondrial matrix, and they also contains mitochondrial subunits and particles. So one by one we will see here. So this is a single crista, ultrastructure of crista, a single F1 particle. So this all these are the different parts of the mitochondria. So one by one we will see here. Now see, first of all we will see mitochondrial membrane. So each mitochondrion is bounded by two highly specialized membranes that plays a crucial part in its activities. So each membrane is a typical unit membrane, an outer protein layer, middle bimolecular layer of lipid, and an inner protein layer. So as like our plasma membrane, the membrane of mitochondria is also a typical unit membrane. So each membrane is about 60 to 70 angstrom thick. Functions of both of membrane, they are different, means inner as well as outer membrane, they plays different functions. So we will see in detail. The mitochondrion consists of two compartments or chambers, namely outer and inner chamber. The space between two membranes, it is known as outer chamber or intermembranous space or perimitochondrial space. So this membrane, it is narrow, and this space, it is narrow and 40 to 70 angstrom in width, and it is filled with watery fluid rich in enzymes. So you can see here, this is the three-dimensional view. Here if you see, this mitochondria, it is made up from two unit membranes. So here if you can see this membrane, this outer membrane, this is known as outer membrane, and this is known as inner membrane. So this is inner membrane, and the space between these two membranes, it is known as intermembranous space or perimitochondrial space. So this is the perimitochondrial space, and this perimitochondrial space, it is narrow and it is filled with watery fluid, and it is rich in different types of enzymes. The space enclosed by the inner membrane is called as inner chamber or inner membranous space. So it is larger than outer chamber and filled with gel-like dense granular matrix material. It is known as mitochondrial matrix. So if you see this all part, it is known as the inner mitochondrial chamber. So this is inner chamber. So this is outer chamber. So this is outer chamber, and this all, it is the inner chamber or inner membranous space, we can say it. And it is composed of what it contains? It contains gel-like dense granular matrix. It is known as mitochondrial matrix. So you must have understand, this is the outer membrane, this is the inner membrane. This part between two membranes, it is known as outer chamber or inner membranous space, and this part, it is which is enclosed by the inner membrane, it is known as the inner chamber, or it is also known as the inner membrane space. So as compared to outer chamber, this inner chamber, it is large and broad, and it is filled with gel-like dense granular material. It is called as mitochondrial matrix. So this is all about the membranes. The inner membrane has finger-like enfoldings projecting into the matrix of mitochondria, and they are also known as crista. So singular, it is known as crista, and all inclusive, they are cristae. or mitochondrial crest. In general, crest of plant mitochondria are tubular, while those of animal mitochondria, they are lamellar or plate-like structure. So you can see here, these are the structures. So these are the finger-like enfoldings which are extended into the matrix. So these are nothing but the cristae. So this singular, it is known as crista, and all these are known as cristae. Cristae may form a complex network by branching in the inner chambers. So here you can see the network like structure which is formed due to these cristae, and this crista provides increased surface area within the mitochondria for various type of enzymatic activities. So due to this crista, a large area it is available for mitochondria for different types of enzymatic activities. The oxidative activities of mitochondria are related to cristae. So oxidative activity means synthesis of ATP or the process of respiration, it will takes place due to these crista. The side of the inner membrane facing towards the matrix, it is called as M side. So you can see here the side of the inner membrane which is facing toward the matrix, it is known as M side. So this is the inner membrane, and this side of the inner membrane which is facing toward the matrix, it is known as M side, while the side which is facing outside, that is toward the outer chamber, it is known as cytosol side or C side. So this is C side and this is M side of the membrane. So here you can see these are two different sides of the membrane. Now second part of the mitochondria we have to see, that is mitochondrial matrix. So this is also very important part of mitochondria. The inner compartment, it is filled with matrix. Now we will see all these part which is enclosed by this inner membrane. So this part, it is known as mitochondrial matrix. Now we have finished mitochondrial membrane, this membrane, intermembranous space and inner membrane space, that is matrix. So now we will see what is the matrix and what it includes or what are the constituents of the matrix. So this entire part, it is known as mitochondrial matrix. So the inner compartment which is filled with matrix which contains a dense proteinaceous material with highly dense granules. Matrix present in the central or inner chamber of mitochondria, it is generally viscous. So this matrix, it is generally viscous and it is semi-solid in nature, and it is composed of fine granules of about 300 to 500 angstroms. So you can see here green color spots, these are nothing but the granules. So these granules are 300 to 500 angstroms. So matrix contains some granules of size 300 to 500 angstrom in diameter. Also this matrix will contains lipids, also it contains proteins and 2 to 6 circular DNA molecules. Also it contains 70s ribosomes. So here you can see these are the mitoribosomes. Here you can see mitoribosomes, you can see this is DNA which is known as mitoDNA. So all these are the parts of the DNA, this matrix, or these are the different types of substances which are present into the matrix. Means it contains some granules, lipids, proteins, DNA molecules, 70s ribosomes, transfer RNA molecules, that is tRNA. So all these are the different constituents of the matrix. The granules consist of insoluble inorganic salts of divalent ions like Mg++ and Ca++. So magnesium and calcium ions can be seen into the granules, and they are very important to accelerate different types of metabolic activities of the mitochondria. The circular DNA molecules are about 4.7 to 5.4 micron long and 20 to 50 angstrom in diameter, and they may be free in matrix. So here you can see this is the mitochondrial DNA molecule have been shown. So this is somewhat this DNA molecule. It is 20 to 50 angstrom in diameter. The enzymes of Krebs cycles are also present into the matrix. So this matrix also contains enzymes of Krebs cycle. Now, third part of mitochondria, it is known as mitochondrial particles. So if we go to observe the ultrastructure of mitochondria, you can see these mitochondrial particles. So the outer surface of the outer membrane and inner surface of inner membrane are covered with thousands of small particles, and the particles which are present on the outer surface are always stalkless and commonly known as subunits of person. So here you can see these are subparticles. Here if you see, these are the crista, and this crista, if you see the enlarged section of this crista, you can see structure like this. So this crista and it contains F1 particles on it, and this crista is 60 angstrom unit membrane. So here you can see this is 60 angstrom unit membrane, and this crista contains such type of particles. Now if you see the ultrastructure of this crista, you can see here. So this outer surface of outer membrane and inner surface of inner membrane, it is covered by these particles. So here you can see all these are the different particles. So here you can see these are the F1 particles. You can see here this is. So inner membrane also contains these particles, and outer membrane also contains these particles. If you see the particles which are present on the outer surface are always stalkless and commonly known as subunits of Pearsons. So these are the some particles which are present on the outer membrane, and these outer membrane particles are always, they are stalkless and they are known as subunits of Pearson. Their number ranges from 10,000 to 1 lakh per mitochondria. So in a mitochondrion, the particle number, it will be range from 10,000 to 1 lakh per mitochondria. The inner surface of inner membrane contains stalked particles. So here you can see these are the stalked particles we can see here, and which are known as subunits of Fernandez and Moran, or they are also known as elementary particles or F1 particles, or they are also known as oxysomes or electron transport particles. So if you see, this is entire three-dimensional structure of mitochondria, and very important part of these mitochondria, these are nothing but these particles, because these are highly involved in the process of the ATP synthesis, or they are also known as oxysomes because they are related with the respiration of the cell or the mitochondria. So these F1 particles are the functional particles which are present into the mitochondria. So these F1 particles are placed at regular distance of 10 nanometer, that is 100 angstrom, and are about 84 angstrom in diameter. So you can see here all these particles, you can see, so all these are the different particles, and they are about 10 nanometer apart from each other, means they are placed at a regular distance of 10 nanometer from each other. If you see the enlarged single particle, you can see like this. So this is enlarged single particle, and which is very important functional unit of mitochondria. Now the number of F1 particles per mitochondria depends upon the size and type of the mitochondria, and generally their number ranges from 10,000 to 1 lakh per mitochondria. Now each F1 particle, what is its function, we have to see. Each F1 particle is composed of 3 parts, that is a round head. Here you can see this is a round head, and this is of 75 to 80 angstrom. Then second part, a stem or stalk of 30 to 35 angstrom in diameter. So this is the stalk, 30 to 40 angstrom, and 30 a stem or stalk of 30 to 35 angstrom in diameter and 45 to 50 angstrom long. So this is diameter is 30 to 35 and its length is 40 to 50 angstrom. Then this F1 particle also contains a cuboidal base and it is formed of 30 to 40 angstrom wide and it forms a part of inner membrane. So here you can see this is a cuboidal base of the F1 particle and it makes the part of inner membrane. Means all these cuboidal bases, they are embedded into the inner membrane. Now the F1 particles, they are meant for ATP synthesis. So this is very important function of F1 particle, because in mitochondria these are the functional unit and they are actually involved in the process of ATP synthesis, that is phosphorylation, and also for ATP oxidation. They are also responsible for electron transport chain. The head of the F1 particle contains the enzymes ATPase and ATP synthetase. So the function of these particles of mitochondria, it is nothing but the ATP synthesis as well as electron transport chain, and they contains different types of enzymes like ATPase and ATP synthetase. Now, if we see chemical composition of mitochondria, the chemical composition of mitochondria, it varies in different plants and animal cells. Mitochondria contains 65 to 70% of proteins, 25 to 30% lipids, 0.5% RNA, and small amount of DNA. Now, lipids of mitochondria, they are composed of 90% phospholipids, and all these different phospholipids, they majority contains lecithin and cephalin. Then it contains up to 5% cholesterol and up to 5% free fatty acids and triglycerides. The inner membrane contains a special type of lipid known as cardiolipin, which is responsible for making membrane impermeable to variety of ions and small molecules like sodium, potassium, chlorides, NAD, AMP, GTP, and so on. So inner membrane contains this is cardiolipin and it is very responsible for making this membrane impermeable for different types of ions. Now, enzymes in mitochondria. Mitochondria contain different types of enzymes. First of all, we'll see enzymes. Outer and inner membranes and mitochondrial matrix, they contain different types of enzymes in it. First of all, we will discuss about enzymes which are present in outer membrane. So, outer mitochondrial membrane, it contains different types of enzymes which are responsible for mitochondrial lipid synthesis. Certain enzymes which are present in outer membrane, they are monoamine oxidase, rotenine insensitive, NADH cytochrome, C reductase, kynurenine hydroxylase, and fatty acid CoA ligase. So all these are the enzymes which are present into the outer membrane. Now in inner membrane there are different types of enzymes present, because initially we have seen functionally both membranes are different or they play different types of roles. Why? Because they contain different types of enzymes. Now inner membrane and inner membranous space, or we may call outer chamber, it contains different types of enzymes. So these are the enzymes in outer membrane. Now these are the enzymes in intermembrane space or the outer chamber, or it is also known as space between two membrane, that is inner membrane and outer membrane. So this intermembrane space contains enzymes like nucleotides, adenylate kinase, and nucleoside diphosphokinase. So several enzymes that use ATP molecules to passing out of the matrix to phosphorylating other nucleotides, they are present into the intermembranous space. Now Next, if we see enzymes which are present into the inner membrane. So enzymes of respiratory chain, electron transport system, and enzymes of oxidative phosphorylation, they are present into the inner membrane. The enzymes of this membrane includes succinic acid dehydrogenase, ATP synthetase, nicotinamide, adenine dinucleotide, flavin adenine dinucleotide FAD, four cytochromes like cytochrome B, cytochrome C, cytochrome A, cytochrome A3, then phosphorylating enzymes, carnitine fatty acids, acyl transferase, ferrochelatases, xylitols, dehydrogenases, etc. So all these are the enzymes which are present into the inner membrane of mitochondria. Then enzymes which are present into the mitochondrial matrix. So these are the very important enzymes which are very functional. The mitochondrial matrix contains a mixture of hundreds of enzymes, and those are required for oxidation of pyruvate and fatty acids and for the citric acid cycle, that is Krebs cycle. So these enzymes are malate dehydrogenase, fumarase, citrate synthetase, ketoglutaric dehydrogenase, isocitrate dehydrogenase, pyruvate carboxylase, crotonase, acyl dehydrogenase, etc. So all these are the enzymes which are present into the mitochondrial matrix. Now beside these enzymes, a mitochondrial matrix also contains several identical copies of mitochondrial DNA, special type of 55S mitochondrial ribosomes, tRNAs, and various other enzymes which are required for the expression of mitochondrial genes. Now, in short, we will see what is mitochondrial DNA, and after that we will see what are the mitochondrial ribosomes. So, mitochondrial DNA, also known as mtDNA, it is relatively small, circular and simple DNA. So, as compared to nuclear DNA, it is small, simple and circular. The size of mitochondrial DNA, it is larger in plants than animals. Its size ranges from 3 micrometer to 30 micrometer, and the mitochondrial DNA is present in the mitochondrial matrix and attached to the inner mitochondrial membrane. The duplication of mitochondrial DNA is under control of nuclear DNA, and enzymes required are imported from cytosol. These mitochondrial ribosomes are the another very important part of the mitochondria, which makes them as an autonomous body, because these ribosomes plays very important role in the process of protein synthesis, and 70s ribosomes which are present into the mitochondria, and they are similar to the ribosomes which are present into the bacteria or prokaryotic cell. So these mitochondrial DNA and mitochondrial ribosomes, they are very responsible to make mitochondria as the autonomous bodies of the cell. Now we will see last part of the mitochondria, that is functions of mitochondria. So mitochondria, they play very important functions. First is cellular respiration and ATP generation. So the most important function of mitochondria, it is cellular respiration, and during which carbohydrate, fatty acids and amino acids are broken up and oxidized, and energy get released into the mitochondria. Now the energy that released is stored into the form of ATP molecules, and thus ATPs are energy rich molecules, and they are generated in mitochondria. Hence mitochondria, they are rightly known as powerhouse of the cell. Then mitochondria along with stored ATP molecules move to the part of the cell where actually energy is required. Means these mitochondria are energy providers. So in cell, they can move from one place to another place where energy is required. So they start, or they will continue liberating energy where it is required into the cell. When the mitochondrial membrane contracts, the inner hydrostatic pressure increases, and due to which water and ATP molecules, they squeezed out of the mitochondria, and they are provided for the different types of metabolic processes. Now when the last phosphate group of ATP is released, a large amount of energy, that is 7,000 calories, is released, which is useful for the different metabolic activities of the cell. Now, the end product of glycolysis, we know very well, in cytoplasm is pyruvic acid. Now, this pyruvic acid undergoes complete oxidation in mitochondria to generate ATP molecules. So, the complete oxidation of pyruvic acid in mitochondria, it takes place by 3 steps. Oxidative decarboxylation, that is oxidation of pyruvic acid. Next is Krebs cycle, that is also known as TCA cycle. And third is electron transport system and oxidative phosphorylation. So all in this way, the complete oxidation of pyruvic acid will be takes place by the mitochondria. Now, next function of mitochondria is role into the lipid metabolism. Now, mitochondria, they are capable to oxidize fatty acids, means they can break down fatty acids, during which energy is released. When the cell is getting starvated or starving, then the oxidation of lipids or fats occur in mitochondria, and it is responsible to produce energy, and so that the mitochondria will remain survive. Then mitochondria are also involved in the synthesis of fatty acids, a reversal of fatty acid oxidation. Means they are responsible for lipid metabolism, means synthesis of fatty acid as well as degradation of fatty acids. Now, role in gametogenesis. So mitochondria plays very important role in the formation of male gametes and female gametes, that is sperm and eggs preparation or formation. During oogenesis, that is egg formation, mitochondria are responsible for vitellogenesis, that is yolk formation. In spermatogenesis, sperm formation, also mitochondria are responsible for proper development of sperm. Now, role in protein synthesis. As we know, mitochondria contains their own DNA, then it contains its own ribosome. So they are known as semi-autonomous organelles. They also contain rRNA, tRNA, mRNA, different types of amino acids and different types of enzymes. And all these are forms of protein synthesis machinery. So mitochondria plays very important role in the process of protein synthesis, but under the control of nuclear DNA. Now, next function is role in extracellular cytoplasmic inheritance. So mitochondria do have their own genetic material in the form of circular DNA, and that DNA play very important role in the cellular inheritance, as they control some important traits of the organism. For example, self-sterility in maize, peptide mutants in yeast. So all these are the different types of traits which are controlled by mitochondrial DNA. Now, next is role in heme synthesis. So these mitochondria of the cell, they are forming, or mitochondria of red blood corpuscles, RBCs, and liver cells store excess quantity of substance called ferritin. This substance is responsible for hemoglobin formation in the developing RBCs, and in this way mitochondria are responsible for the synthesis of hemoglobin in RBCs. Now, next is role of mitochondria in gluconeogenesis. So the gluconeogenesis is the process of formation of carbohydrates or conversion of non-carbohydrates into pyruvic acid to glucose. It is known as, non-carbohydrates such as pyruvic acid to glucose, means the conversion of non-carbohydrates into carbohydrates. It is known as gluconeogenesis. So mitochondria, they play very important role in the process. Next, role in amino acid metabolism. So the enzymes for oxidative deamination of amino acids are present in mitochondria, and these enzymes includes glutamate dehydrogenase, proline dehydrogenase, etc. So all these enzymes are very important for the amino acid metabolism, and it is carried out by mitochondria. Last function, if we see, it is heat production. In some hibernating animals, the color of fat it becomes brown due to high concentration of mitochondria. So in such animals, heat is generated by the ATP of mitochondria, and all these animals they are able to survive in that condition also. So these are some common functions of mitochondria. Apart from these functions, mitochondria plays very other important functions also. So hope you understand about mitochondria. Thanks for your attention. If you like video, share, comment and subscribe the channel. Thanks for your attention.